ever wondered how scientists extract DNA from an organism? All living organisms have DNA. It is basically the blueprint for everything that happens inside an organism's cells. We study DNA for many reasons. To figure out how the instructions stored in DNA help our bodies function properly, to make new medicines or genetically modify crops to be resistant to insects, and even to solve crimes and reconstruct evolutionary histories. And the first step for all of this is the extraction of DNA. In this activity, we will make our own DNA extraction kit from household materials and use it to separate DNA from Kiwi. The first thing that we need to do is to prepare the extraction buffer. An extraction buffer is a solution that will help to break the cells in order to extract the DNA in a process known as cell lysis. To prepare our extraction buffer, first, mix 90 ml of distilled water with 1/4 tablespoon of salt. Distilled water serves as the solvent for our buffer. Salt decreases the DNA solubility to water as it neutralizes the negative charge of the DNA. In effect, this will cause the DNA to clump together, making it easier to isolate the DNA. This process is called salting out. DNAs are also called in proteins called histones. Salt removes these proteins in order to free the DNA. Next, add 10 ml of clear hand soap into the solution. Our cells are composed of fatty acids, which binds to the molecules of the soap. Since soap is soluble in water, it makes it possible for fat to be dissolved in water. Mix the solution well, but be careful to avoid foaming. Lastly, measure 20 ml of the solution. This will be used in our experiment. Now for the DNA extraction, peel 2 to 3 kiwi fruits with a knife and slice them into cubes. Using a mortar and pestle, mash the kiwi slices. In this process, we are trying to break the fruit into smaller pieces in order for us to easily extract the DNA. Put the mashed fruits into a clear jar or a glass. Then add the 20 ml extraction buffer solution. Mix it well. Separate dinner bowl, prepare a warm water bath. Fill half of the container with hot water and half with tap water. Once the water is comfortable to the touch, put the jar containing the kiwi mixture and let it stay for about 10 to 12 minutes. This process aids in cell lysis and it also degrades and deactivates some of the proteins which may contaminate the DNA extract. After 12 minutes, remove the Kiwi mixture from the warm water bath and subject the jar onto an ice bath for 5 minutes. Subjecting the mixture onto an ice bath will help prevent DNA degradation after being subjected to heat. For every minute, remove the jar and mix the solution. After 5 minutes, remove the jar from the ice bath. Then, filter the large cell debris onto a funnel with cheesecloth in order to get the kiwi extract. Take the ice-cold ethanol and pour it slowly on the side of the jar with the extract. Let the solution sit for 2 minutes. This process is known as alcohol precipitation. Ethanol aids in the precipitation process by making the DNA less soluble to water as it gets attached onto the salt molecules, increasing the yield of the DNA. For 2 minutes, you will start to see a slimy white mucus substance starting to form on top of the solution. You can scoop it using a paper clip. This is the DNA. So why did we use kiwi as our source of DNA? It is because the cultivated kiwi fruit is a hexaploid organism with 174 chromosomes. The volume of DNA is directly proportional to the ploidy level. Hence, the higher the ploidy level, the higher the DNA yield. Kiwi has a relatively large ploidy level, so we are expecting to extract a large amount of DNA.